And Reza, I'm going to hand over to you if I may, and let's go. Thank you.
Thank you very much, David, and thank you for the Royal Free Hospital Kidney Patient Association for inviting me to give this talk. Can I just check everyone can hear me and see my slide? Perfect. Okay. So if you don't know me, I'm Reza Motallebzadeh. I'm a kidney transplant surgeon and academic based at UCL with my lab in the Division of Surgery and the Institute of Immunity and Transplantation, this nice new shiny pears building that you see at the front of the hospital. And one of my main interests is in transplant immunology, in particular, how the gut immune axis can affect kidney transplant rejection. So I'm going to give you a bit of background to the work and give you the sort of hot off the press, the latest data we have from our study. So, oops. Yeah, just a reminder about the amazing success of kidney transplantation. This is looking at patients who survive compared to patients on a waiting list who have either a deceased donor or a living donor transplant. And as you can see, a transplant for those patients who are eligible and can get a kidney transplant provides tremendous not only quality of life, but improvement in years of life compared to waiting on the waiting list on dialysis. And however, when we look at the graft failure or graft attrition rate in different years of transplants, so if we look in the early 1980s, so late 1980s, early 1990s, you can see that the 1 year attrition rate was about just over 16%. And due to the massive improvement in immunosuppression, medical and surgical techniques, that attrition rate, that 1 year attrition rate is now down to about 4%. However, if you look at the mid and long-term survival of kidney grafts, so looking at 1 to 3 or 3 to 5 or 5 to 10 years, we haven't really made that much improvement. This is still sort of bumbling along at 4% attrition or graft loss per year. If you look at through this different decades of transplantation. So this is an area of an improvement that we really need to make an impact on in order to improve the long-term survival of kidney transplants. If we look at graft failure, what are the main causes of graft failure? Well, unfortunately, one of the biggest causes is dying with a functioning graft. So the kidney graft is working, but there are other causes of death, such as cardiovascular disease or infection. But the second largest cause of graft failure is this alloimmune pathology. This is 25%. And that alloimmune pathology reflects immune injury in the form of rejection to the kidney graft. And that can limit the long-term lifespan of a kidney graft. And this is US data. In the US, this is not that long ago, from 2018, 50% of kidney transplants are lost by 10 years. And that means patients going back on dialysis and a significant cost to healthcare systems. So in the US in 2016, this accounted for $35 billion. Well, when you looking at our own composition, what are we actually composed of? Well, actually, we're composed more of non-human. We're actually more non-human than human. In fact, we're superorganisms. So we're composed of 39 trillion microbial cells, so outweighing the number of human cells, which are 30 trillion. And if we look at our sort of genetic composition, we're composed of 23,000 human genes, but actually 2 million microbial genes. And so actually, when we consider any immune response, we have to consider not just our cells, but our microbes, because they form a significant proportion of our bodies. And as you will have heard, this composition of microbes, called the microbiome, which exists within surfaces of the skin or in the respiratory tract or in the gut, have been linked to various diseases. And the changes in the composition of the gut microbiome has been known to influence both cerebral disorders such as neurodegenerative disorders, autism and psychiatric disorders, It has a link to the lung, in which there's been links to changes in the gut microbiome, has been linked to things like lung cancer. Metabolism, very important, the gut microbiome in controlling things like obesity and diabetes, and also cardiovascular disease. And because of this strong interaction between the human gut microbiome and the immune system, that really influenced my ideas or thoughts on how this can play a role in how the gut immune axis can play a role in kidney transplantation. If you look at the sort of taxonomic tree, the significant differences between the microbiome between the in the gut of adults and children. So why is the role of the gut microbiota important? Well, the gut of healthy adults is populated about over 1 trillion bacteria. Its microbiota tends to be defined by the body site. So what you have, the microbiome in the gut, is very different to the microbiota, for example, in the oral cavity or on the skin or on the respiratory tract. They're important for driving the immune system. So if you don't have a microbiome, you don't develop an immune system. They orchestrate the development of lymphoid structures. So these are things like your lymph nodes and spleen, which are important to fight infection. And they control maturation of effector and regulatory responses. And also there is a sophisticated crosstalk between the microbiota and the host immune system and metabolism. So if you have changes in the gut microbiome, you can potentially see changes in things like disease processes such as diabetes and certain forms of cancer. So as the microbiome plays an important role in training and maintenance of the immune response, we can see that in fact, multiple diseases, even outside the gut, are influenced by the type of the gut microbiome and how it influences immune cells. And as I mentioned, things like diabetes, even brain disorders such as multiple sclerosis. There have been links of the gut microbiome influencing the type of T cells that can influence development of arthritis and also certain forms of allergies. So it's not a leap of faith to think that, well, if you have a patient who's receiving a kidney transplant who gets different types of drugs such as antibacterial, antimicrobial prophylaxis in the form of antibiotics, immunosuppression, and surgery, this can cause a potential change in the type of the gut microbiota. And maybe this can influence the type of the immune cells that the microbiota interacts with to influence either rejection and fibrosis of the graft, or in fact, actually can result in tolerance or improved allograft function. So if we think about chronic kidney disease, we know that the gut microbiota are important to break down fiber, which releases these important metabolites such as short chain fatty acids and tryptophan metabolites. And the interaction of these bacteria on things like dietary fibers, which we need the gut bacteria to digest, and the release of these metabolites can influence different populations of T and B cells. And those T and B cells can actually be effector cells, so they can be used to sort of fight infections and viruses, but they can also be regulatory lymphocytes, so act as a brake on the immune system to prevent an immune response from getting out of control. And we know in patients with kidney disease, there is this massive disruption of these types of gut bacteria, so that you have lower microbial diversity. There's changes in the gut barrier function, so that the lining of the cells lining the gut are a lot more leaky. And this results in potentially bacteria moving across the epithelial surface and into the systemic circulation, which can result in upregulation of inflammation, downregulation of these sort of regulatory responses, and the tip of an imbalance between the degree of effector lymphocytes and regulatory lymphocytes. So you have more active immune response and less regulation. So our hypothesis is that in patients with kidney transplant, patients who undergo kidney transplantation, there's reduced gut barrier function because of the type of drugs and because of the disease process that is the cause of their end-stage renal failure. This leads to changes in the gut microbiota and metabolites that can control immune responses, so that you have an imbalance. You have less regulatory lymphocyte populations, which dampen immune responses, and more effector lymphocyte populations, which promote immune responses. So our aims of our work was follows: firstly, to assess, well, what is the gut barrier function like? Is it normal or intact in patients who undergo kidney transplantation, and does that leakiness I described earlier, does that resolve? Secondly, to characterize what are the type of bacteria within the gut microbiome and the metabolites that they're producing. Thirdly, to profile the type of T and B cells, the lymphocytes that are in the peripheral blood that are important to regulate immune responses, and in particular will be important in determining whether a kidney transplant is rejected or not. And fourthly, to determine associations between the type of metabolites that the gut microbiota is producing in these in patients undergoing transplantation and immune cell populations. So we have undertaken a study of just under 100 kidney transplant recipients and about 20 live donor patients at the Royal Free Hospital. And we've collected blood, urine, and stool samples before transplant and at various time points after transplant. So first month, 3 months, 6 months, and 12 months. And our main outcome measure of the study is to determine which patients have this biopsy-proven rejection, whether they've developed a rejection episode or not, which has been confirmed on biopsy. And there's lots of things that we do with these samples. So for the fecal and urine samples, in this particular study, I'm just going to focus on the fecal samples. We genetically sequence the DNA within the fecal samples to determine what are the type of species of microbes that are present in within the gut. And we've done this work previously in pediatric patients undergoing stem cell transplantation. Our blood samples we put through a machine to determine the type of cell markers present on B and T cells. And thirdly, we measure gut permeability. We assess gut permeability by measuring the level of this intestinal fatty acid binding protein, which is a protein specifically released by gut lining epithelial cells. And then fourthly, we assess the type of metabolites that the gut microbiota is producing within the gut by using a very fancy machine called a mass spectrometer, which can analyze a host of different proteins within both the serum and fecal samples. So this was our baseline characteristic of our participants who kindly enrolled into the study. So we had 83 kidney transplant recipients and 20 live donors. And we chose live donors as healthy people who are, as a control, who are undergoing a major operation, but not receiving immunosuppression, but still also receiving 1 dose of antibiotics. And you can see that they're well matched in age. The median age was about 53 in the recipients and 56 in live donors. We had predominantly deceased donor patients who underwent a deceased donor transplant, 70% versus 30% live donor. And then if you look at this figure here, just under 16% of patients had an episode of rejection, of which the majority we call T cell and B cell mediated rejection. So the first thing was to assess the gut barrier function. And as I've mentioned earlier, that when the gut lining is injured, it releases this protein called intestinal fatty acid binding protein, which we can measure in the blood. And really interestingly, what we see we measure these levels of IFAB from before transplant, this time 0, and then at 1, 3, 6, and 12 months. And really interestingly, we can see that patients who go on to develop rejection have a much higher level of IFAB before their transplant compared to patients who do not reject their kidney. And then after the transplant, these levels normalize to what's very similar to sort of healthy control or live donor nephrectomy patients. So the next step was to characterize the gut microbiota and their metabolites. And we did this by, as I said, sequencing all the sort of bacterial species within the fecal samples. And we have this measure called alpha diversity, which measures the range of richness and evenness of the gut microbial communities. And you can see that after transplantation, there's a profound loss of diversity. So the gut microbiota becomes less diverse. There is some recovery after the transplant, about 3 months to 6 months, but it never really goes back to what it was prior to transplant. Although there's this profound change, we couldn't really see a difference between patients who reject or who don't reject in terms of their composition of the microbiota. Again, this shows that in patients compared to live donor nephrectomy, patients that got live donor nephrectomy, at 1 month there is this profound drop in diversity of the gut microbiome, and that could be as a result of the surgery, as a result of the immunosuppression, and a result of the antibiotics that we give. And then if you look at how far apart they are from compared to at different time points, so you can see that this is what we call a principal component analysis. Just very simply, it gives an idea of how far the gut microbiota is changed compared to baseline. And you can see most of the gut microbiota is here. And in more detail, we can see that actually there's loss of certain anaerobes. These are types of bacteria that require a limited amount of oxygen. So we see that there is a profound loss of anaerobes within the gut microbiota in patients after transplantation. So what does that mean in terms of metabolites? So these, as I mentioned, these gut microbiota are important in generating metabolites. There's no other way of generating these metabolites without the gut microbiota. And in homeostasis in health, one important metabolite is this molecule here called indole-3 acetic acid. It's an antioxidant and binds to this AHR receptor, and it's involved in promoting regulatory cells, in particular T regulatory cells. And then another type of metabolite, 5-HAA, which is also derived from tryptophan, so from things that you eat, like certain fruits and vegetables, they can promote expansion of B regulatory cells. And both these tryptophan metabolites can act on types of antigen-presenting cells and can also restore gut barrier function. So when we looked at tryptophan levels after transplantation, we've separated these again into patients who don't have rejection in blue, patients who have rejected their graft in red, and live donor patients who've donated their kidney in gray. And as you can see, for both the patients who don't reject and reject, there's increases in tryptophan after the transplant. And that's probably because that patients have moved on to a less restricted diet once their transplant is up and running and working. So even though that there's more of this tryptophan, however, the metabolites derived from tryptophan, such as indole-3 acetic acid or 5-HA, these are two important metabolites which are broken down by the gut microbiota acting on tryptophan. We see that in patients who reject. You can see at the time point of rejection, which is predominantly at 1 month, you can see that those levels are profoundly reduced. And similarly, the other important metabolite, this isobutyrate, is also reduced at 1 month after a transplant in patients who have rejected their graft. So something is happening that, although we can't see a difference in the microbiome, on the gut microbiota, in the types of bacteria, we are certainly seeing differences and drops in levels of these important metabolites that are important for gut barrier function, but also for regulating immune responses in patients who go on to reject their graft. So how does that tally with the type of T and B cells we see in the peripheral blood of our cohort of patients? Well, if you look at T regulatory cells, this is one of the most important cells in your body that are critical to controlling and dampening down immune responses. So So immune responses don't get out of hand, and you don't develop things like an autoimmune response, and certainly they're important to limit the development of rejection. You see that patients who have rejected their graft, there are predominantly lower levels of T regulatory cells compared to patients who should display no evidence of rejection. And another type of regulatory cells, these are called T follicular regulatory cells. These are a specialized subset of T regulatory cells which control antibody responses. And again, at 3 months, you can see that there's a profound difference in reduction in these level of T follicular regulatory cells that control antibody-mediated rejection. And these are much lower, and so therefore less able to control, limit the rejection response. And we see that quite clearly at this time point. So finally, what I was going to just show you, just a very small snippet of data to look at the associations between these important metabolites that are reduced and these lymphocyte populations that are critical in controlling immune responses. And what you can see, this is what we call a heat map. So the red squares represent if there's a strong association, and blue squares represent the sort of an inverse association. And if we look at our regulatory subsets of cells, which we've highlighted here, and if we look at the different types of metabolites, just 3 to point out, there's indole propionic acid, 5-AHA, and serotonin. These are correlated with presence of regulatory subsets. So, this is really quite novel data in kidney transplantation that we've now shown that metabolites derived from tryptophan are positively correlated with T regulatory cell subsets. That means that the more of these metabolites you have, the more regulatory subsets or T cells that your body can generate. So what conclusions do we have from this work? Well, firstly, we've seen that there's increased gut permeability in patients who go on to acute rejection, and this increased gut permeability is evident at the time point before transplant. So there's obviously potentially some degree of inflammation that's going on that's determined that is driving, say, an immune response potentially to promote acute rejection. And that transplantation results in a loss of diversity of the gut microbiome and loss of certain gut obligate anaerobe. And certain metabolites, so these produced by the gut microbiome, such as the 5-HIA, indole-3 acetic acid, are positively correlated with these proportions with peripheral numbers of T regulatory cells and T follicular regulatory cells. These cells are really important to dampen down immune responses. And that if we what this work is implying is that if we restore these key metabolites and if we restore gut barrier function prior to a transplant, we may then be able to limit the development of acute rejection after a transplant. And this is a really a novel way of thinking about how to limit rejection in patients, so that it's not just relying on immunosuppression. We have potentially a whole new therapeutic area that we can apply to kidney transplant patients to limit the development of acute rejection and hopefully extend the long-term lifespan of a kidney graft. So it just remains for me to thank my collaborators. Clearly, I couldn't have done this work without their help and without the tremendous support that the pairs building and the great cohort of scientists that are working with me there, my collaborators at the Institute of Child Health, and most importantly, the study participants who've kindly donated samples and their time to taking part in this really, what I think is a very fascinating study. And I've just listed important funders of the work: the Royal Free Charity, Royal College of Surgeons of England, St. Peter's Trust, and the National Institute of Health Research that have kindly provided funds to support our area of research. Thank you very much.
Thank you very much, Reza. That was fantastic, fascinating. Okay, do we need to leave your presentation?
I can stop sharing, yeah.
Yeah, great, okay, thank you. I've got a few questions that have come in here, and as you are not currently looking after cooking your supper, Diane, you can, if you are going to unmute you or ask you to unmute, you can ask your question.
Hello there. My question is, how do different immunosuppressants actually affect the gut permeability? Personally, I think that MMF, when I first started on MMF, I was going to the loo, emptying my bowels like 4 or 5 times just in the morning. So
Yeah,
I'm wondering about the immunosuppressants themselves.
I think that's a very, very good question. So MMF is notorious for causing things like diarrhea, I'm afraid, and it's really difficult to tease apart whether the MMF is by itself causing these changes in the gut microbiota that we see, or the other immunosuppressants that we're giving, such as the tacrolimus, the steroid, the antibiotics. So it's really hard to unpick that. And clearly, the fact that MMF can cause diarrhea, one would postulate that that's having a profound impact. But actually teasing it apart from the other immunosuppressants is quite difficult to do in terms of is it that which is particularly causing the change in the gut microbiota or something else. So yeah, it's a challenge to unpick that one.
Thank you.
Thanks, Diane. Alexander, I've lost the chat now. Are you able to? Yeah.
Yeah, I can ask my question. This is Alessandro Bortoni. I was transplanted mid-January, so it's slightly less than 3 months. Yeah,
Hi, Alessandro. I know you well. I did your transplant.
Ah, you did, exactly. Sorry, you got
Yeah.
you got the light behind. I couldn't.
Yeah,
see
it was me.
and I
Yeah.
didn't recognize your name. I'm very sorry. You're
That's
right.
all right.
Yeah. Now I recognize you. Well, thank you very much. But I had a very bad urinary tract infection. Let's say a little bit less than a month after, and I was treated with many antibiotics and so on. And now I'm well now, but I still have some bacteria in my urine samples, not too high level. I don't have symptoms, but then I'm given antibiotics, they go away. They stop the antibiotics, those sachet every day you take every 3 days,
Yes.
and then they come back. So I come to the conclusion that since I've heard and I've read that it happens, that those bacteria pass from the bowels to the blood, and that's the of permeability. I believe that since I had 2 surgeries, so all the antibiotics that I've taken, they must have destroyed my gut flora. And for this reason, I decided by myself, but then I told the doctor, the nephrologist, and he agreed to take probiotics. So my simple question would be, since you mentioned to restore it before the transplant, what can we do after the transplant to restore gut diversity? Because even if it's not rejection, it is still quite annoying, because I would like to travel and go on holiday, and the last thing I want is to have a urinary tract infection with fever or cystitis and so on.
Sure. That's a really important question, Alessandro. So a couple of things there. I mean, actually, what we're also interested in, part of the research, and we haven't analyzed our data yet, is to look whether actually if there are genes within the gut microbiota which are sort of antimicrobial, so that, and these genes can potentially be passed from the gut into the sort of urinary system. And it's possible that actually a lot of urinary tract infections that we see after a transplant, and especially if they're sort of antibiotic resistant, they could possibly be harboring within the gut microbiota. And actually those antimicrobial genes, we're at the moment doing a very sort of detailed analysis to see whether patients who've developed a urinary tract infection, whether they've harbored these antimicrobial resistance genes within their gut microbiota. And as you quite rightly point out, that if you take antibiotics, there's very good evidence to show that you change the diversity of your gut microbiome, and having a more diverse microbiome is somewhat protective of certain forms of infection. So So anything which you can do in terms of things like a, probiotic to try to restore that, especially when you're taking antibiotics, is probably a good idea. Although there isn't really strong evidence for that in kidney transplantation, and that's where sort of our work is hopefully going to go next, is actually try to, now that we've seen these associations, just to try to restore that gut microbial diversity prior to a transplant. And there are many different ways of you can do that, either with a prebiotic or a probiotic. You might have heard these things called fecal microbial transplants.
Yeah.
This is usually used for things like if you've got intractable Clostridium difficile diarrhea, C. diff, and it hasn't been trialed yet in kidney transplantation. So I wouldn't recommend that, because there's side effects from that, and there's no evidence to suggest that they are of benefit. But from the data that we can see, it does imply that having, potentially restoring that gut microbial diversity, restoring that gut microbial function and barrier function is potentially protective for your kidney transplant.
Okay, because the doctors are quite interventionist, they make any sort of hypothesis like, oh, we're gonna go in with a cystoscopy, or maybe it's your native kidneys, we need to remove them. So obviously before doing that,
Yes,
I'd rather try,
yes,
a smoother approach, also because
yes.
I believe it comes from that. But anyway,
Yes.
Thank you very much for your answer and for the surgery.
No, you're welcome.
Thank you both. Esther, you've got a couple of questions. So if you unmute yourself, you'll be able to present them.
Yes, well, you've answered the fecal transplant question, which is that's one thing. So in practical terms, what can we do to enhance is there, like Tim Spector, who is another expert, he says try and eat 30 different plants, and he said cocoa is a plant and spices are a plant. He said try and eat 30 a week, but that's not really talking about probiotics and prebiotics.
Yeah.
Is that something that we can just send off to Amazon for and add that in?
I think where sort of this is going to be sort of become really in terms of game changing is to have it personalized. So one would need to know what is your own sort of gut microbial characteristics, what is there, and what needs to be potentially restored in terms of, or are we seeing a change in your or drop off in certain metabolites that we know are protective for example, for a kidney transplant, or protective at least for the immune cells that control rejection. So I think I think if you're taking antibiotics, it's not a bad idea to take a probiotic of some sort, so that you know that you're trying to sort of preserve the diversity. I couldn't recommend one or another. But I think that's in fact, a very close relative of mine had to take antibiotics, and she was given a probiotic to take at the same time because there's very good evidence that does disrupt your gut microbiome. Where we're lacking, and this is where our work is leading to clinical trials to see, well, can we personalize the probiotic or prebiotic to an individual to restore the key metabolites that we think are important for controlling things like kidney graft rejection or urinary tract infections.
Okay.
Can I just say that I've been taking this. It's a remedy which comes from Arab doctors, and my GFR has gone up from 15 to 21 in the same number of months. So 1 GFR per month, and it's red onions chopped with honey. 3 teaspoons in the morning and 4 late afternoon on. So 7 teaspoons of it a day.
Right.
It's not fun, but I keep saying to myself, it's better than all those pills I would have.
Right.
Transplant.
Right. Yeah. Yeah. It'd be interesting to see to sequence your gut microbiota from before to afterwards to see what changes have, if any, have happened, and whether that's the the cause of your change in GFR. As I said, what one person's gut microbiome is very fundamentally different to another person's. So whilst our own microbiome can be relatively stable, unless we have things like antibiotics or immunosuppression, there's a massive degree of interpersonal variability. So your gut microbiome will be probably fundamentally different to mine, and therefore having a very sort of blanket treatment, it's difficult to prescribe because of those differences. And I think personalizing the type of pro or prebiotic to an individual is really going to be where the work can be translated into something quite meaningful on a population level.
So in other words, that it could stop somebody from needing a transplant, or it could stop them from needing dialysis.
I mean, I think I can't really say that because that's not what our work has shown. But what we have shown, we've seen these associations between the degree of, say, gut inflammation and development of rejection. We've seen these metabolites that are important in controlling immune responses that can impact whether a kidney graft is rejected or not. So, based on this work, it would seem plausible that, well, if you restore your gut barrier function and if you restore some of these key metabolites, and it's knowing how to restore those metabolites, not just by giving them, but maybe by influencing the type of species of bacteria that's in the gut, on the gut lining, then it's potentially possible that will lead to less rejection. Whether those same metabolites are important for things like chronic kidney disease, that's a different area of work, and I wouldn't want to speculate on that. And lots of people have actually done this work in chronic kidney disease, and it's shown that there are certain metabolites which are less frequent. And again, it's pointing to a lot of kidney disease is autoimmune disease, so your immune system reacting against itself, and that's because maybe there's an imbalance between the B and T cell populations which are dampening down the response, such as the regulatory cells, versus the effector cells which are promoting a response, similar to what we see in rejection. And there are some parallels between autoimmune disease and transplant rejection, between these balance of these subsets of cells. And it is clearly plausible also that the gut metabolites are also having that same effect. But just want to point out that our study wasn't looking at autoimmune disease, particularly just focused on transplant patients.
Do you recommend reading any of the kinds of researchers that have been preventing dialysis, preventing kidney transplants? Do you recommend any reading for that?
When you say preventing, what do you? Well,
like for me, honey and red onions has been preventing me from hitting dialysis point. And I just wondered if there was anyone else who's written and done research on it, because I'd be buying their books if there was.
Okay, and I mean, not that there might be one or two research papers that look very interesting that I can share with David, and then he can then pass on to you if that's
Thank you.
Yeah.
Okay.
Yeah.
Thank you, Esther and Reza. Jeremy, I can see your face now. Is your audio working, or shall I do the question? Okay. No. This is Jeremy Gold's question. He's not a transplant patient, but he has vasculitis, which has caused stage 3 CKD. So he takes immunosuppressants to control that. Are there any implications, Reza, in your work for his type of case?
Thank you, Jeremy, for the question. So similarly, as I was explaining to Esther, that in autoimmune disease, again, it's this imbalance between sort of regulatory cells which dampen or put a brake on your immune system versus effector cells which promote an immune response. So in your case, a vasculitis response. And it's possible that this imbalance is controlled by this disruption between the gut and the gut microbiota and immune axis. So I can't comment on vasculitis because this is not work that we've done in vasculitis patients, but it does have implications that similarly would be interesting to look in patients who have an autoimmune disorder. And we know that in diabetes that there are profound changes in the gut microbiota, and whether that it similarly applies to other autoimmune diseases such as vasculitis, and whether again restoring gut barrier function is important for that. And we've one of my colleagues within the PAIRS building, she's done a lot of work on juvenile arthritis, which is again another type of autoimmune disease that affects the joints in children, and they've shown some really nice work that actually the type of the gut metabolites are very important in controlling B regulatory cells. And if you don't have these certain types of metabolites, or if you have a low amount of these metabolites, especially this metabolite called butyrate, which is derived from fiber, those patients have more profound arthritis and more profound SLE also. And she also looked in SLE than patients who have normal levels of these metabolites.
Okay, Jeremy, is that all right? Does that satisfy your question? I can't see you now. I've lost you, but anyway, I'm sure it does. Thank you, Reza. Patricia Gooden, are you still with us?
Yes, I am.
Right. Why don't you ask your question?
Trying to see if my camera will work now.
Don't worry, just ask the question.
Okay. Yeah, I wanted to ask the professor, what criterion did you use to choose the 100 patients in this study?
So thank you, Patricia. So we approached patients who could first of all understand the sort of the basic outline of the project and what it involved. If they were if they're above the age of 18, if they're having a kidney transplant alone, so not a kidney and a liver or a kidney and a pancreas, and if they're obviously willing to donate their urine, blood, and fecal samples at various different time points along their transplant journey.
Okay, thank you very much. Thank you.
You're welcome.
Did you answer this one from Esther? Do you agree that gluten is one of the biggest culprits regarding leaky gut, and that giving up bread would help the gut?
No, I didn't answer that, but I think that's quite contentious. I wouldn't really. I'm not an expert on gluten, but I don't think that's it's really hard data to suggest that gluten is a cause of gut barrier dysfunction, because we'd all be. I mean, there'd be a lot. I think that the evidence is rather selective, I would have to say, based on that, and I probably wouldn't advise giving up bread for everyone.
Okay. Good. Could you recognize the actual makeup of someone's microbiome's breakdown to actually eliminate the future risk of virus and bacteria?
I'm not quite sure. We can sequence what are the microbes within the gut, and we can really get down to quite detailed level of what are the different types of species within the intestinal tract. Why would we want to eliminate them? I'm not quite sure I understand that. Is it eliminating future infections or?
Yeah. Yes.
Yeah. So, I mean, the relationship between certain species and another species within the gut microbiome is quite complex, and there's a lot of interdependency. So if you get rid of one, you will affect potentially other pathways which are important in as I say, in breakdown of fiber. And a lot of these key metabolites come from fiber. And if you don't have the gut microbiota present there to break down that fiber, you will not generate those metabolites. So if you, for instance, if there are certain colonies of experimental mice called germ-free mice, they have a vastly different gut microbiota compared to wild-type mice, and they're more susceptible to certain diseases, or they can't than the sort of wild-type. So it is quite a complex interplay, and I think if you try to sort of remove one, you're at risk of limiting development of certain metabolites that are really quite essential to human health.
Yeah, yeah. So really, there's, though we've been talking about some bad elements of microbiome, there really actually a lot of good ones. Like as I understand it from what you've been saying, they obviously help you digest food and actually protect against infection.
Correct. Yeah. So important that a lot of them are key for maintaining the gut barrier epithelial lining. And if you have a breakdown of that epithelial lining, you have more systemic inflammation. There's links to cardiovascular disease. So it's just quite important that that lining is intact, and that only happens if you have sort of a wide, diverse gut microbiota. And again, certain metabolites are important in controlling your immune system. They can only be produced by gut microbiota. There's nothing else that can do that, as long as you have the right diet as well.
Yeah, yeah. And tell me, as a last question here, do they play a part, or are they affected by COVID? Or do they have an impact on COVID?
Yeah, so those immune cells, those metabolites that I mentioned that control different populations of immune cells, those are clearly important also for certain forms of infection, or combating infection. So and it was seen that patients who developed severe COVID had a different makeup of their gut microbiota. There's quite good evidence for this compared to patients who recovered well. And again, that's pointing to the fact that whilst in a transplant, you want to have higher numbers of regulatory immune cells to dampen down your immune response so you don't get rejection. In infection, it's the other way around. You want more of your effector cells and less regulatory cells, so you produce a good response, immune response to combat the infection. So it is somewhat contextual depending upon what it is that you're trying to sort of manipulate. Are you trying to promote, prolonged graft survival, in which case you want metabolites that are limiting the immune response, or are you trying to fight an infection, in which case you want certain forms of metabolites that are going to promote differentiation of effector immune cells to clear that infection?
Right. Thank you for that. Caroline, you've got a question. Welcome.
Thank you, David. I do realise this is a very speculative question. I mean, acupuncture is a treatment which can promote balance in the body. It's known for that, and it's also known for conditions relating to pain. I just wondered if anybody got any evidence that acupuncture might be helpful in balancing the gut biome.
Wow.
Yeah,
I mean, to be honest, I have not come across that research in that area, so I don't want to comment.
I get that. There is a reason I'm asking, actually, because a long time ago, when I was suffering from kidney failure, I did actually have acupuncture quite regularly. One reason was, like, say I was in pain from gout, it would just get rid of the pain. But the other reason was to try and balance my body as well as I could. And I don't know if that was the reason, but my kidney failure was incredibly slow. I mean, who knows?
Right. Well, I mean,
but
yeah.
if you're saying now that it's important to have a balanced gut biome, it does occur to me that that is a treatment which is used because it's about balancing the energies in the body. So that might actually be something that might just help. So I thought I'd just mention that.
Yeah. No, it's interesting. I haven't come across I haven't come across that, so I don't really want to comment.
No, I get
Yeah.
Caroline, did the acupuncture have any impact on the gout pain?
Totally, yes.
Really?
Oh, absolutely. Yeah, I don't know how I'd have managed without it, actually, because nobody
Well,
offered me any drugs, and I didn't take very well to having this really incredibly painful big toe. I couldn't bear a sheet on it or anything. No, it was really, really helpful. It took the level of pain down massively, and the pain went fairly quickly.
well, I'll have to get your acupuncturist's name and
Well,
details off
unfortunately,
you
this is in
because
Newbury. I don't live in Newbury. I might be looking for one round the North London area, actually. I
yeah,
don't have
well,
one.
I mean, my guy is around that area and he's well known, but he didn't ease any pains in my body. But anyway, maybe your guy, you paid more. I don't know.
Well, it
Anyway,
was a woman.
all right. Okay. I'll pass on to you the name of my guy.
Oh, thank you very much, actually. I would really appreciate that. Thank you, David. That would be
Okay,
great.
all right. Jill, you had a question, and I'm going to have to say that this is the last one because we're just gone a few minutes over 7 o'clock. Jill, can you ask your question?
Yes, actually. Yes, thank you for your presentation. Some of it I didn't understand, but I'm wondering if I missed the fact that I thought when I first was transplanted, I've had my transplant for 15 years now, that you weren't meant to eat live bacteria, food with live bacteria in it, such as probiotic yogurt. So because I understood it sort of could cause infection or inflammation.
Yeah.
So I sort of avoided it, but I noticed that people are talking about eating probiotics.
Yeah. So, Jill, thanks for your question. So, I mean, there have been some trials using probiotics in certainly in liver transplantation, and there is some evidence, not very strong, to show that there was reduced infection rates in patients who took probiotics after their liver transplant. So that it has been used. I mean, they have to be, obviously, these are highly regulated. And most of them contain things called bifidobacteria, and they're safe. I mean, those ones that they use in those trials. The prebiotics are the things which are undigested, sort of non-digestible foods that can stimulate the growth or activity of certain microbes. And now a lot of there's one clinical trial actually in Australia which has just started in transplant recipients by giving something called inulin, which is a prebiotic, and that's again designed to stimulate the growth of things like bifidobacteria or lactobacilli, and it's potentially that trial is looking at whether the metabolic profile of patients after kidney transplant, so less cardiovascular disease, less diabetes, whether that can be influenced by these prebiotics. But again, that's a trial which has just started. So again, there isn't really what I call hard evidence in terms of a randomized controlled trial, whereby you have one group of transplant patients who have a prebiotic and one group of a placebo, and whether that's going to actually influence any outcomes.
What is a prebiotic in terms of food exactly?
They're sort of it's difficult to describe. It's sort of they're non-digestible foods. I mean, by all means, there's one of them is something called inulin. They're not particularly tasty foods, I must say, but they're designed to sort of stimulate certain growth of bacteria.
So it's food that contains inulin, you mean?
Well, the substance is something called inulin.
Oh.
Yeah, that's a type of prebiotic.
Right, you can't buy it over the counter.
Actually,
Amazon.
yeah. Okay.
Thank you.
You're welcome.
Thank you very
All right.
much.
There's a couple of other questions which I'll send to you, Reza, by
Sure.
email, and if you'd be kind enough. So, ladies and gents, I'm going to wind up this part first because I think Nee does want to say something very quickly. So, Reza, you can see by the amount of questions that we've thrown at you that whilst I'm sure for myself and a couple of other people that have put in the chat that they will need to watch this again to try and understand, because it's, you know, there's so much in it which is a little bit foreign to us, but it's been fascinating. So thank you so much for doing it. And you and I will see each other in other meetings in the future. Everyone, even if you want to give a soft applause to Reza, thank you so much.
Thank you. Thanks, David. Thank you, everyone. Really enjoyed that. Thank you.
Bye. Bye-bye.
Bye-bye.
Thank you.